Welcome you to this webinar, Comprehensive DIA Proteomics with Thermo Fisher Orbitrap Astral from Sample to Insight. Before we get started, let me just give you a quick reminder of our webinar guidelines. First, all attendees are automatically muted upon joining, so there is no need to adjust your microphone. Secondly, we will have a live Q&A session after the presentations. Please type your questions in the chat box at any time, and we will address them during the Q&A. And finally, this webinar is being recorded. If you would like a copy of the recording, it can be requested after the session. With that, let's move on the brief introduction of Novogene. Novogene is a global leader in multi-omics and is a global leader in genomics and multi-omics services. We are also listed in Shanghai Stock Exchange. Now let's take a quick look at our global footprint. We operate 10 laboratories and 19 subsidiaries worldwide, with major hubs in Singapore, China, US, and Europe. This global network allows us to deliver rapid turnaround times, maintain strong quality control, and provide local support wherever our customers are. In the EMEA region alone, we are proud to serve more than 400 accounts and support over 10,000 projects every year, spanning across 42 countries. Here is an overview of our multi-omics services and solutions. Our portfolio covers genomics, transcriptomics, including single-cell sequencing and spatial transcriptomics, epigenetics, microbial sequencing, and of course, proteomics and metabolomics. Together, these services allow us to support comprehensive biological research from multiple angles. So now, I'm pleased to hand the session over to Dr. Dawa from Thermo Fisher. He will introduce us to the Orbitrap Astral platform and share how this technology is shaping the future of proteomics. Dr. Dawa, yeah, the floor is yours.
Yeah, thank you, Wendy, and good morning, everyone. It's my pleasure to share about Astral mass spectrometer technology. So let me share my screen. Wendy, you need to probably enable me to.
Yes.
Okay. Let me know if you can see my screen.
Yes.
Okay, yeah. So again, yeah, welcome all. Thank you for joining this webinar in partnership with Novogene. So what I'm going to do in this 25 to 30 minutes presentation, I will introduce what Astral MS technology is and then give you some example data. And at the end, I will also showcase how we are taking this technology forward and what is the impact that it is making in the research field. So before that, I have 2 slides to introduce Thermo Fisher. I think Thermo Fisher, we define our mission to enable our customers to make the world healthier, cleaner, and safer. And this is the purpose, what we do every day and why we do what we do to serve you better. And it is a global leader when it comes to life sciences solutions. And we are having industry-leading brands that supplies boundaries of innovations and support many different solutions. And we have a global expertise that you could rely on at over 120,000 employees with significant investment in R&D to bring cutting-edge technology to meet the unmet needs for the research. With this introduction, I will move on to the presentation. We are talking about proteomics. So this slide captures the translation research. We believe that proteomics has a huge potential to unlock the true potential of biology. And if you look at the field over the last 15 years or so, there is significant work has been done in terms of genomics. More than 1 billion genetic variants have been identified, right? However, we realized that purely using genomics alone, we cannot explain the disease etiology as well as pathophysiology of many diseases. Some statistics, probably hardly 15 to 20% could be explained based on genomics. That's why people want to look at proteomics, right? When you move from genomics to proteomics, the 20,000 genes might lead to more than a millions of protein variants, right? Because of post-translation modification, protein-protein interaction, and so on. So that's why it's very important to study proteomics to unlock the true potential of human biology. And these days, there is also a lot of talk about multi-omics. And then I'm not sure if you have heard this terminology called proteogenomics, which is basically integrating genomics, transcriptomics, and proteomics data to aid the discovery as well as quantification of proteoforms. And to emphasize again, because genome alone cannot explain the underlying progression of many diseases. However, protein abundance can be impacted by genetic variation, ancestry, as well as exposures, right? So that's why it is very important to integrate these different fields into multi-omics via proteogenomic studies. And just to a quick introduction, right? Orbitrap-based mass spectrometry is gold standard when it comes to proteomics. And there are some unique informations that you could obtain from mass spectrometry-based proteomics that is not possible with other alternative technologies. For example, is unbiased interrogation, identification, quantification, that is also possible with other technology, but you could also go further understanding the dynamics, protein-protein interaction, and post-translation modification that is only possible by using mass spectrometry-based proteomics. However, there are several challenges when you want to move mass spectrometry to the genomic scales, right? So let's say if you want to do proteomics is the scale of genomics, we still lack throughput because current platforms do not have sufficient throughput to do a population scale proteomic studies. We still lack the sensitivity if you want to go deeper, and if you're doing single cell level work, we still need a higher sensitivity to go even deeper. So we know that these are the challenges, and we work over the years to overcome these challenges by coming out different types of mass spectrometers. And this is the instrument that was used to do a human proteome mapping. I think this was the first commercial Orbitrap mass spectrometer. It took them several years, and as you can see, instrument is bulkier, is slower. And since then, we have several iterations of Orbitrap mass spectrometry. And if you're talking about translation research, proteomics, multi-omics, and if you just zoom into those high impact journal publications, tier 1 journals such as Nature and Science publication, we can see that over the years, Orbitrap has huge impact on scientific outcome, as we can see with exponential growth of publications over the number of years, which highlights the importance of this technology in scientific world. And as I mentioned, we are continuously innovating to improve it further. Then lately, we also introduced many different workflows, right? So if you identify protein, it doesn't give you a biological insight unless and until you add the quantitative impact. So it's not only important to identify, but also to quantify protein to give to understand the biological impact. And these are, I would say, rather three different major workflows that are supported by Orbitrap mass spectrometer, whether you are using data-dependent acquisition or data-independent acquisition, whether you are doing any labeled or label-free, whether you are using our TMT multiplexing by labeling using TMT Pro reagent, or you are using targeted quantitation using parallel reaction monitoring or PRM. So all these workflows has been supported by all versions of Orbitrap platforms. So as I mentioned, we are continuously innovating, right? So since the first commercial Orbitrap in 2005, this year we are celebrating 20 years of commercialization Orbitrap of mass spectrometer. And in 2023, we have a huge leap in commercialization of instrument by launch of Orbitrap Astral mass spectrometer, which is essentially designed to meet the unmet needs, particularly for proteomics meaning achieving higher throughput, high sensitivity, and deeper coverage simultaneously. And just to highlight the impact that this particular technology has made, within the first year of launch itself, we have 44 publications, which is much more than what we had with any of our platform that we launched previously. And just yesterday, I was checking, and now over 2 years and 3 months since we launched, we have now 500 over publication, which shows the impact that this technology has made in a scientific world. And to my knowledge, in my last 15 years, there is no other analytical platform that has so much research outcome in first few years of commercialization, right? We have 500 over publications in just slightly over 2 years with this particular platform, because it is able to meet the unmet need that researchers were looking for all this year. So that's why we believe that this is the most significant advancement for mass spectrometry in last 15 years. And those 500 publication, if I dissect, this is just a snapshot. I think I did not update this matrix for a while, but it just shows diversity of applications that this instrument is able to cater, from single cell to bulk proteomics, and beyond proteomics to metabolomics, and beyond human proteomics to plant proteomics, and so on. Right? And the instrument is good enough, but whether it's a robust and stable, right, is also important, particularly if you want to democratize it to do a large cohort studies like what we have been doing with genomics. So this data shows that over 250 days of continuous operation, we maintained quite robust performance over 24,000 injections, right? So this shows that instrument is not only high throughput, very sensitive, but it's also very robust, enabling you to do a large cohort studies without any worry. So I just want to introduce this parallel processing, right, before I go into how Orbitrap Astral technology works. If you are familiar with Arsenal of Venice, in the 16th century, this is one of the most comprehensive naval base, because they could make one ship per day, right, by doing 100 different processes in parallel. This was in 16th century. If we compare with modern car plant, now this modern car plants can manufacture 100,000 cars per year from a single plant by doing parallelized and pipeline manufacturing, right? So what they have been doing, the production line move units between processing stations in parallel. And by doing this parallel processing, they increase the productivity and efficiency by reducing the overhead in all the processes. We have been trying to do the same thing with different generation of Orbitrap mass spectrometer. With Astral, we have taken this to a next level. So what it is, right, as I mentioned, this is the most significant advancement in last 15 years. It is a combination of 3 different mass analyzer: the quadruple mass analyzer, the Orbitrap mass analyzer, and Astral mass analyzer. So this is the only technology where we have 2 high resolution mass analyzer working in tandem and in parallel, right? And by doing so, we achieve a new standard of mass spectrometry performance, and I will show you later on. We are able to do a 5 ions packet parallelization by combining this 3 different mass analyzer into one box. And this gives us throughput, sensitivity, and coverage that we need to do large cohort studies or to go deeper for single cell proteomics. So this is how the schematics looks like. So now a few slides I have a bit on the hardware. So if you are not much into mass spectrometer, you may find it difficult to understand, and I will try my best to simplify it. So this is how the schematic of the Orbitrap Astral mass spectrometer looks like. This is the ion source where we convert the sample into gaseous form, ionize, then we go into the quadruple mass analyzer where we select the masses of interest. And then we have the Orbitrap mass analyzer. So this is the same as our Explorer 480 with mass resonance up to 480,000. And the big change is the back end where we have this Astral mass analyzer. So if I zoom in further to the Astral mass analyzer, we have several innovations here. We have an ion processor that can do a scanning up to 200 hertz, so very fast fragmentation and data acquisition speed, combined with high dynamic range detector and innovative technology to reduce the ion losses when we do MS/MS data acquisition while maintaining 80,000 resolution, which is significant when it comes to MS/MS data acquisition. Even with Orbitrap mass analyzer, typically we use 15,000 to 30,000 mass resolution we are doing MS/MS, but with Astral, particularly for MS/MS analysis, we can do up to 80,000 mass resolution. So now we are combining the high resolution of Orbitrap for MS1 scan and MS/MS2 capability of Astral mass analyzer into a one box. And by doing so, right, this is how the Orbitrap, Astral mass analyzer looks like. You have a asymmetric ion mirror, and then you have third dimension with ion foil, which controls the drift of the ions, right? And by doing this unique architecture, we minimize the loss of ions. So that's why the Astral stand for asymmetric track loss analyzer. And why it is important, right? If I just show you some of the statistics, right? If looking at, this is just lipidomics data, just a small molecule compared to Orbitrap base MS data acquisition, the Astral mass analysis gives you more than tenfold improvement in sensitivity, mass resolutions, and everything is tenfold higher when it comes to MS2 data, right? And because of this, we get very high sensitivity, high speed of data acquisition up to 200 hertz, at the same time maintaining high resolution and low ppm mass accuracy. So this unique combination allows us to achieve unparalleled performance. So now I will show you how the parallelization process works. So again, this is the ion source, this is a quadrupole, this is the Orbitrap mass analyzer for MS1 scan, and these are the Astral mass analyzer for MS/MS2 data acquisition. So first, we will have a Orbitrap mass analyzer, which will do a full scan analysis. And after doing a full scan analysis, the ions would go to the ion routing multiport, where we can do ion concentration as well as precursor isolation. And then after that, it will go to the ion processor, where we'll fragment the ions to do a fragmentation. And those fragmented ions would further go to the Astral mass analyzer to do MS/MS data acquisition. And then they will be ejected into the high dynamic range detector to generate a spectrum. And as I mentioned, at one point of time, we are doing 5 parallel processes. So the first process is in Orbitrap mass analyzer, right? We will do the full scan analysis. And while it is doing a full scan analysis, we will accumulate the ion in ion routing multiports, and accumulated ions would be further fragmented into ion processor and then accumulated into this ion optics, and then would be ejected into Astral mass analyzer. So you have 1, 2, 3, 4, and 5 simultaneous processes happening in parallel. And because of this, we are able to achieve very high acquisition speed and throughput, right? As I mentioned earlier, in 1 second, we are able to achieve 100 MS2 spectrum and 1 full scan MS, just to show you how it works, right? Sorry. Okay, right. So this is what it does in real time, right? This is how it will continuously generate a spectrum. If I compare this to real time, to this simulation, we have to ramp it up by 56 times faster to have a real-time performance, right? But it's incredibly fast while maintaining very high resolution while acquiring the data. Right, and because of this instrument also allow us to do what we call a narrow window DIA. So this is a classical DIA, and this is a normal DIA. When I say normal DIA, we are using 50 to 100 Dalton window, whereas with narrow window DIA, we are using 2 Dalton window width, right? And again, this diagram below shows the comparison. So DDA, of course, we have less coelution, but it's not sensitive enough because we are only doing more like targeted approach. DIA, it has a higher precursors, but then there is a lot of co-elution, as you can see in this yellow band. So narrow window DIA is a combination of both. It gives you improved sensitivity and selectivity over a DIA window. And why we are able to do so? Because of incredibly fast scan speed, we can now do narrow window DIA on Orbitrap Astral. And by combining all these features, what we get is a faster throughput, a deeper coverage, very high sensitivity, and simultaneously we are getting very accurate and precise quantitation. Now, with this introduction of technology, I will show you some of the data very quickly, right? So when we introduce this technology, we enable analysis of 8,000 protein groups in just 8 minutes, right? So if we compare this is like 4 times higher throughput than the best available technology before the launch of Orbitrap Astral, right? So now you can do much more faster. And it's not only faster, right? We can go even deeper, right? So if you compare with previously based technology, in order to do 12,000 proteins, we can now do it in 60 minutes compared to only 8,000 proteins. And if we compare a throughput, right, we can have, as I mentioned, 4 times higher throughput. So different run times, right? Even if in 15 minutes run time, we are able to achieve 8,000 proteins. If you run it longer, 1 hour, right, and you can even go deeper. So now we are able to do a single sort proteomics by using Orbitrap Astral very quickly, right? To just show you, I will show you that slide later. I mentioned about TMT multiplexing. So the Orbitrap Astral also support 18-plex, meaning you can combine 18 sample into a 1 sample and do 1 injection to analyze 18,000 sample by using a TMT-based labeling, right? And this is just to show you in previously, if you want to do a complete proteome coverage, you need to do 46 fractions, and then you introduce each fractions one by one, so which would take you around close to 35 hours in order to achieve 12,000 proteins. But with Astral, you can achieve 12,000 proteins in 1 hour, but if you extend a little bit longer, you can now do a 15,000 proteins over 270 minutes runtime. So now we are enabling single sort proteomics in few hours compared to few days by using this particular technology. How about plasma proteomics, right? So the plasma sample is one of the challenging samples. So even with neat plasma, with 60 samples per day, we are able to achieve 700 protein IDs. If you do depletion, we can go up to 2,000 proteins with 24 samples per day. And these days, there is a lot of different strategy to do sample enrichment. If you do enrichment, so we partner with Sphere. So with Sphere enrichment technology, we can go easily 6,000 proteins with 30 samples per day. And this is the data about 2 years ago. Recently, Sphere also come out with a new protocol where you can double the throughput. Now you can do 60 samples per day with about 6,000 proteins coverage using Astral as well, right? So now you are able to do very high throughput plasma proteomics enabled by Orbitrap Astral MS. How about single cell proteomics, right? So people have been doing a bulk proteomics. However, the bulk proteomics does not give you a very accurate information for heterogeneity of particular tissue or organism. So if I give you example of such as brain, right, is very heterogeneous. There is a difference between different cell populations. So that's why there is a lot of interest to do a single cell proteomics. However, to do a single cell proteomics, you even need very high sensitivity. And now we are analyzing a single cell, so throughput is even more important because otherwise it would take you forever to complete one particular sample, because in one particular sample, you might have to analyze 80, 100 or few hundred cells, right? So if you do several injections, it will take you forever. And why it is important, the sensitivity, as you can see, right, depending on the cell of your interest, they comes with different size. So you are loading very minor amount. So that's why the sensitivity is important to do proteomics coverage. And this slide shows the workflow. I will not go into a detail. I think some of the workflow would be covered by Novogene presentation later on. But we have our Vanquish Neo with microwave column, easy spray source, and FAMES Pro. This is again unique differential ion mobility device as a source that can be added to Orbitrap Astral. This helps you to remove +1 and other charge states so that you can improve the sensitivity. And this is particularly important for single cell work, right? Then combined with Orbitrap Astral, and you can use our own or other available data processing software. And you can see, compared to what has been achieved by other platform technology, we can nearly achieve 2 times higher throughput with 4 times higher coverage. So now we are able to achieve 80 samples per day with 5,000 plus proteins, even at single cell level sensitivity. So this is again enabling the throughput and sensitivity that was not possible before. And we did a comprehensive comparison as well, right? So this is another method by loading a 250 picogram of HeLa cells across our different laboratories, and while maintaining over 5,000 proteins, close to 6,000 protein groups, we can still maintain a very robust performance with less than 2% RST across 3 different sites using the same methodology and on 3 different instruments. This is our own data, and then in a literature, people are even pushing it further. So this is again another publications where they demonstrated almost lossless single cell proteomics workflow with a throughput of 80 samples per day. And not only that, in the same experiment, they were also able to do a post-translation modification even at single cell level, right? So again, this incredible performance achieved with Astral as a platform. I mentioned about accuracy and precision. So identification is meaningless if it is not accurate and precise. So this just shows that when you do a 3 proteome mix, and even with different CV threshold, we are able to achieve a very good quantitation and nearly close to expected ratio. So what it shows that instrument is also very precise and accurate in terms of quantitation for complex samples. And we can also have a higher coverage, right? So compared to this blue, the gold standard Orbitrap platform with Orbitrap Astral, we are able to achieve a larger dynamic range, meaning you are able to uncover more trace level protein biomarkers from your samples. And with metabolomics, right, I talk about proteomics. I have just 2 slides to show you that instrument is also able to do metabolomics and lipidomics studies. So we have been talking about this workflow called simultaneous quantization and discovery, where we want to combine targeted and discovery workflow in a single injection. So what we can do, you can have your label standards or standards run concurrently together with your samples. And then we will do both targeted as well as untargeted analysis by processing data differently from a single injection. And when we do so, compared to Astral based platform, right, we can achieve a higher throughput, right? Even in 15 minutes gradient, we are able to achieve a high percentage of features. Even we scale down to 5 minutes, we can still maintain more than 90% of MS2 fragmentation. And if we compare together with Orbitrap platform, so the 5-minute run on Orbitrap Astral is equivalent to 15-minute runtime on Orbitrap based platform. So again, showing you that this technology gives you a firepower to go deeper and confident unknown identification, even for metabolomics, lipidomics, and small molecule applications. But as I mentioned, we doesn't stop here, right? We continuously strive to innovate further. So I have a few slides to introduce what we have done. So earlier this year, about a few months back, we introduced another family member in Orbitrap Astral. We call Orbitrap Astral Zoom mass spectrometer. So this platform is built upon Orbitrap Astral technology, but this is now the highest performing instrument when it comes to proteomics, and the second is still Orbitrap Astral. Both platform allows you to do single cell to bulk proteomics, but with Orbitrap Astral Zoom, we have made it more versatile to do metabolomics, lipidomics, biopharma, and it has a novel methods such as hybrid DIA and enhanced dynamic range mode and so on, all possible to do both targeted and unbiased discovery in a single injection. So again, this is a schematic of Orbitrap Astral Zoom. The major difference compared to Astral is this. Now we introduce this band trap, right? So another step for pre-accumulation. So in Astral, we were doing 5 ion packet parallelization. With Orbitrap Astral Zoom, we have 6th. Now we can do 6th ion packet parallelization. So by using this band trap, we can do a pre-accumulation which enable us to achieve 40 to 50% higher ion abundance with Orbitrap Astral Zoom. And then we have slight modification on Astral mass analyzer, so enable us to do 32 plex, and it can go even faster, 270 hertz. If you're doing a small molecule analysis, now it has enhanced dynamic range mode to enable it to do even 6 order of dynamic range. And now we introduce hybrid DIA, in which we can do both PRM and DIA simultaneously in a single injection. So several improvements to take it further. And in terms of performance, as we can see, when we launch Astral, it's already break a world record by covering 8,000 proteins in 8 minutes. But now we can do over 8,000 proteins in 5 minutes, meaning we can now nearly double the throughput with Orbitrap Astral Zoom. And why we are able to do so? Because we have increased speed to further. Now it can go 270 hertz, and combined together with pre-accumulation step, we can even achieve a higher throughput beyond Astral. So that's why I like this phrase, accelerating the accelerated. And this is some additional data. Again, all this data we are comparing with Astral because there is no other platform that can come close. So as we can see on this diagram here, when we go different sample per day, right, the higher is the throughput, you will see a better higher difference. So ranging from 5 to 15% improvements in protein group as well as peptide coverage, moving from Astral to Astral Zoom. And at the same time, we are able to maintain very good reproducibility. So this is a median CV, less than 4% at protein level and around 10% at peptide level. So it's incredibly great performance, improving further on what we could achieve on Orbitrap Astral. So this data using 200 nanogram of HeLa digest. I have another set of data, right, to show you. And this is data from Jesper Ostland. As we can see, previously, when we launched Astral, we were able to do 180 samples. Now with 300 samples per day coverage, we can do 300 proteomes per day kind of throughput. So nearly we are going into the high throughput space that can be matched with what we can achieve with genomics based with technology. And you can see if you at 300 protein samples per day, we can have a similar performance with Astral using 180 days. Sorry, 180 samples per day performance. How about single cell? So we even introduced this mode called low input mode by some modification in detector. So now even we can go 5% more improvement in proteins groups as well as the peptides. So even achieving higher and giving you a more confidence when you want to do low input mode. So if you do select the low input mode on our Orbitrap Astral Zoom, it will automatically align the detector to detect low input mode, as we can see here, by improving the intensity, and that's what results in higher coverage and better data at a single cell. And this is again, previously when we did the similar data, we break a world record by having 5,200 proteins on this particular single cell. Now with Orbitrap Astral Zoom, we can go up to 5,800 protein on a true single cell. So even further improving the sensitivity for single cell level as well. How about plasma proteomics? So as I mentioned, with a new workflow from Seer, with 60 samples per day, we can go up to 6,000 plus protein, both on Orbitrap Astral and Orbitrap Astral Zoom. It's just that with Astral Zoom, you will have 10 to 15% more coverage of protein. But now we can even go beyond 7,000 if you would run 24 samples per day kind of throughput. So really incredibly higher sensitivity paired with the throughput that can be achieved with Orbitrap Astral and Astral Zoom, even with complex samples such as plasma. Okay, last slide, last data on immunopeptidomics. So, recently there has been a lot of interest to do immunopeptidomics. So there are many different diagrams here. The first shows that even with different loading material, we can maintain the high range, high dynamic range across different loading amounts. And we can see we can go from 9,000 to close to 28,000 peptide coverage, depending on amount of the sample that you load. And while we can achieve a higher coverage, we are not compromising the spectral quality. Even with E5 to E8, we can maintain the spectral quality. So now is even able to enable you to do high sensitive, high throughput immunopeptidomics using Orbitrap Astral mass spectrometer. And what it has done, so this slide shows you, right? I've shown you a lot of the data. And remember earlier I said that genomics is mature technology. People can do large cohort studies, more like routine cohort studies. With Orbitrap Astral launch, now we are able to do mass spec-based proteomics at population scale. And these are some of the large cohort studies that we can see globally. People have been utilizing the Orbitrap Astral to do a different cohort ranging from 10,000 to 50,000 samples. So now it is possible to complete 50,000 samples in few months, which was not possible about 3 years ago. So now we are in the range of throughput comparable to genomics, which would enable you to unlock the human biology by doing population scale proteomics. Last data, right? I did not cover this, but if you want to do a spatial biology or spatial proteomics, we have been also working with different partners to do spatial proteomics. So in this case, this is the data from Stanford University, where we combine with fluorescent microscopy, where you do cell isolation using immunoprecipitation. Then you do a laser directed isolation of the cells, and then you inject those cells into an Astral mass analyzer to do mass spec based proteomics. And then you can overlay your fluorescent microscopy with mass spec data to do spatial proteomics, right? If you are interested to know more about, I think reach out to us and we can provide more information. So in summary, the Orbitrap Astral instrument family incorporates a new technology that combines the sensitivity demanded by single cells to the speed and dynamic range for bulk proteomics as well as multi-omic studies. It gives you a fastest throughput, deepest coverage, highest sensitivity, and simultaneously giving you very accurate and precise quantitation. Right? And beyond Astral, we also have a range of the instrument, whether you are doing a targeted omics using your Stellar, or if you want to do a structural biology post-translation modification using our tribrid platform such as Orbitrap Ascent, right? And beyond LCMS as well, Thermo Fisher, when it comes to chromatography and mass spectrometry, we have solutions to virtually analyze any inorganic or organic analyte, ranging from LCMS, ion chromatography, GCMS, ICP-MS, you name it, we have the solutions. So no matter what is your analyte interest, we do have solutions to analyze that in any given sample, right? With this, I would like to thank the team who were involved in innovating this incredible technology, as well as our customers and collaborators from industry and academia who were very incredibly supportive to come out with these innovations, and my colleague from whom I borrow some of the data that I presented today. And thank you, and I'll be happy to take any questions towards the end of the presentation. Yeah, thank you.
Thanks, Dallas. A comprehensive presentation regarding the Orbitrap Astral, the data performance, and the applications. So next, if you got any questions, please feel free to type your questions on the chat box, and we will answer later. So next, I will move to Dr. Yue Bai, who is product manager for the proteomics from Novogene. Yeah, welcome, Yue Bai. Over to you.
Okay. Can you hear my screen?
Yes.
Okay. Good morning. It's a pleasure to be here today. I'm John Yuebai from Norwegian. Please excuse my camera being off. I'm on a business trip, and the connection isn't stable enough for video. Today, I will walk you
Sorry,
through.
could you use the full screen? Yeah, now it's still the present model.
Reliably detect over 10,000 proteins per project in different cohorts. Even with tricky samples like human bone marrow and plant tissue, we still get satisfactory results. This really demonstrates the strength of our workflow and its power for large cohort study. Let's look at a specific application, blood proteomics. Plasma authoring is information-rich sample, and low abundant proteins in blood samples are likely to be potential biomarkers and drug targets. The feature of these special samples are complexity and wide dynamic range. All this significant disturb mass-based detection. Therefore, a variety of methods for blood sample pretreatment have been developed. There are mainly two types, depletion and enrichment. The representative of depletion is antibody-based kit, like thermal depletion resin. The representative of enrichment is nanoparticles. Nanoparticles are defined by their physical and chemical properties, so they are not limited by species. With the upgrade of instruments and preprocessing methods, the in-depth analysis of proteins in blood sample become possible. Uh, we can see both depletion and enrichment methods lead to a significant increase in number of identified proteins. And our strategies maintain a high detection number while ensuring excellent repeatability. As we all know, generating data is only half the battle. The real value comes from interpretation. We provide a comprehensive suite of analysis, including protein functional annotation, protein quantitative analysis, differential protein expression analysis, functional enrichment analysis, GLCA analysis, and protein-protein interaction analysis. All these results place your findings in a biological context. In addition to providing regular reports, we conduct more comprehensive analysis on the blood samples. We annotate protein concentration using human plasma protein project database. This approach suggests that we could prioritize low concentration proteins. They are often the most promising biomarkers. Furthermore, integrating databases like Human Protein Atlas and the Drug Bank, we can provide disease-related proteins annotation and druggable protein annotation. All these results help you to bridge the gap from discovery to therapeutic. Beyond standard pipelines, we excel at customized analysis. Every project is unique, and our team are ready to work with you to develop custom strategies and answer your most specific biological questions. As researchers move forward, we are seeing that proteomics shouldn't look at alone. Proteomics could be combined with other omics to get full biological picture. If we want to truly understand how organ works, we should bring together proteomics with genomics, transcriptomics, metabolomics, and PTM profiling. This multi-omic approach helps us unlock biological mysteries. One area that gets a lot of attention is combining transcriptomics and proteomics. And the genomics and the proteomics are powerful together for finding new biomarkers and drug targets. Proteomics plus metabolomics is used for studying metabolic disease. And when we look at proteomics along with PTM profiling, we get a much clearer view of molecular switches and codes. In Novogene, we can provide multi-layer service, including genomics, transcriptomics, epigenomics, proteomics, and metabolomics. We've got the tool to support your research from multi-angles. Here, I will represent the integrated analysis of proteomics and metabolomics for biomarker screening. Think of biomarker as your body warning lights. They signal that something might be changing or going wrong with us. And biomarkers give us a crucial peek into our health. And the research on biomarkers plays a vital role in improving drug development and advancing biomedical studies. Proteins are the direct executor of biological process, and proteomics can foreshadow what is about to occur, while metabolomics relieves what is currently happening and reflects the molecular outcomes of all biological phenotypes. By integrating proteomics with metabolomics, we can bridge the gap between molecular changes and functional outcomes. This integration offers profound insight into biological mechanisms and provides our capacity for identifying diverse biomarkers. Furthermore, this combination approach allows findings to be cross-validated, providing a more comprehensive and reliable picture of biological system. Here is a case about mass-based biomarker screening for diagnosis. To identify novel biomarkers, proteomics and metabolomics analysis for all serum plus samples from patient and healthy controls were processing. Researchers conduct a random forest to discriminate patients from healthy controls. A time-flawed cross-validation was performed, and the accuracy was evaluated by ROC crew. We can see in protein classification model, 3 proteins are considered as biomarkers. The AUC value for biomarkers combination reached 1 and 0.85 in training and testing data respectively. And using this 5 metabolite combination, the AUC was calculated as 1 in both training and test data. The validation of biomarkers in independent cohort shows that the both of 2 validations obtained high sensitivity and specificity. It means this study has promising potential in clinical situation. However, a key limitation is the use of only a single classifier and evaluation method. This approach can lead to unreliable results because different data types have distinct characteristics, and there is no universal optimal algorithms. Also, even though they look at both proteomics and metabolomics data, and they didn't actually combine them for a more complete analysis. In Novogene, we help you cut through the noise of your complex proteomics and metabolomics data to find a set of biomarkers with highest predictive power for diagnosing a disease or condition. After data normalization, we leverage not 1 but 4 powerful ensemble algorithms for screening. For validation, there are 6 machine learning models and 6 comprehensive evaluation metrics in our workflow. In the end, a diagnostic model is generated by formulating the equation using regression coefficients and interception of biomarkers. This format is user-friendly and practical. And a truly powerful biomarker is defined by its robust performance across a wide range of proven algorithms, not just a single complex AI. This multi-omics validation improves the reliable and stable. This principle is the foundation of our workflow, ensuring the results are ready for the real world. Finally, let's see some cases based on proteomics and multi-omics. This study was mainly focused on the discovery of biomedical materials and mechanisms, and the detection and analysis of proteomes was supported by Novogene. Through animal experiments, it was discovered that tissue bandage could promote agogenesis and regular avoiding behavior and bladder function after MPG injury. Proteomics has elucidated the key mechanisms by which TB treatment exert its effects. Tissue regeneration and M2 macrophage proliferation were the key to functional recovery. Interestingly, many researchers are attracted by this network. This interaction network was generated based on the data and graphing software guidelines from our reports. This is our strategy that combines both professionalism and practicality. This study is about the interaction between environment and fungi. Through a series of biological experiments, the potential of degrading microplastics by edible fungi has been found. The oyster mushroom was regarded as having the greatest degrading ability. Integrated analysis revealed 291 co-expressed genes and proteins, and functional enrichment analysis showed that they are primarily involved in metabolic process, oxidative stress responses, and membrane transport. Further analysis showed that several novel enzymes and metabolic pathways has vital roles in degrading microplastics by oyster mushroom. This study broadened the potential for applying edible fungi as an engineered fungal platform. The last study dives into precision medicine for colorectal cancer. Using a bunch of advanced tools like whole exome sequencing, RNA-seq, and proteomics, researchers developed new treatment strategies for colorectal cancer. RNA-seq and functional screening confirm the potential therapeutic target CDK4. Based on identification and analysis of drug sensitivity, OBA01 and ABN was established as the optimal treatment combination. Besides, proteomics identify the key mechanisms underlying this synergistic effect. Notably, of the two drugs in this study, OBA01 and ABN, OBA01 is a clinical stage compound, and ABN is already approved. This work paved the way for expanding their clinical indications. Just as this article states, an increasing number of applications have proved that multi-omics become a strong tool for biomarker and drug target discovery. For deep profiling to high-throughput screening, Novogenes provides a true one-stop solution for all your needs. Thanks for your attention. I'm now happy to take any questions you may have. That's all. Thank you.
Yeah, thanks, Bai, for the excellent presentation for the one-stop solution from Novogene Proteomics Services. So we are a little bit running out of time. We have two questions here. One is for Dr. Dava. You mentioned that running the sample longer on the Orbitrap Astral can yield more protein data. Is there a maximal runtime for samples, or how can we determine when the protein rates have reached their maximal capacity?
Yeah, thank you. Thank you for the great question. So I think this is not only applicable to Astral mass spectrometer, this is in general. Of course, running longer, we give you a higher coverage, but there is always a compromise that if you run longer, will not get more after certain points, because it would be dependent on two things, right? The data redundancy and stability of your sample. So typically, when we say longer gradient is 60 minutes to 120 minutes, median is 30 to 45 minutes, or shorter gradient like 15 minutes. In order to determine whether, how long is enough, so you can plot the curve of identified protein peptide versus runtime, right? So I think I'll show some data where we do 30 minutes, 60 minutes, 50 minutes, 120 minutes. So you can do a different, generate different data at different runtime, and then you see whether how much protein ID you are gaining. After a while, it will reach a plateau. And sometimes you also need to look at the reproducibility as well. So you need to do this in replicate studies to see whether are you able to consistently get a run or not, right? So, but after a certain point, yeah, it will reach a saturation, right? So there is no limit on the instrumentation, but typically for your sample, yeah, this is how I would determine, how much runtime is enough. Assuming you have ample time, if you're not limited by the runtime, then you can run 1 hour or 2 hour long gradient to see. Other approach which people took it, you can do a sample fractionation, right? Of course, you can split the sample, do a fractionation, and run individual fraction longer. So that's how you can also increase the higher coverage. But if you have the Astral, you don't need to do the fractionation. You can just run 60 minutes gradient. You would have most proteins. And if you run it longer, yeah, you will have almost a near whole proteome you will able to achieve it.
Okay, thanks, Dr. Dawa. Yeah, we do have another question for Yuebai. Where are your specific requirements for sample preparation and shipment? What kind of sample preparation services do you offer? Or do we need to provide ready-to-run peptides? Are there any QC steps we have to do if we were to submit ready-to-run peptides?
Okay, all you need is to do lab your sample with their name, and then you can hand them off to us. For cell pellet, tissue, serum, plasma sample, please send them in a form box with dry icing. For gel samples, please pack them in and include an ice pack before sending. We can handle samples for all types such as tissue, cells, protein, blood, and you name it. We have optimized protocols for each one, so you don't have to worry about processing them into peptides. And for QC steps, we have two types QC steps. First, for instrument QC, we have a regular maintenance plan, including daily quality control sample testing, weekly calibration, and periodic cleaning. For sample QC, after license, protein concentration is quantified using BCA assays, and integrated is assembled assessed by SDS page. In additionally, the digest peptides mixture is analyzed to evaluate peptide level characteristics. The accompanying QC report includes the following metrics: peptide legends distribution, retention time distribution, and the unique peptides distribution. That's all.
Okay, thanks, Yuebai. Yeah, I guess we are a little bit running out of the time, so we will end the webinar soon. If you got any questions, please write to Norwegian directly. So here I put our linking and website. So you can scan the code here and leave your questions to our website, and our sales rep and the TS representative will contact you directly. So before the webinar close, just a quick reminder: at the end of today's webinar, there will be a short questionnaire. We would really appreciate it if you could take a moment to share your feedback with us. Thank you. Yeah. Okay, if there is no other questions, we will end the webinar today. Thank you again for Dr. Dawa and Dr. Yuebai to do the presentation and introduce the services for Norwegian. Hopefully, we can contact again regarding the services customers will ask. Thank you.
Yeah, thank you all for joining. You can reach out to me via email address or via LinkedIn. Yeah, thank you.
Yes, thank you very much.
Thank you.
Okay, see you.